I think we complete the disaccharides. Now we have to go with the polysaccharides. Is that
The polysaccharide also finished.
completed? Okay, so did we go through the significance of carbohydrates?
No, sir.
We have to start from here, no?
Yes, sir.
Yes. Okay. Yeah, the thing that you want to understand from here, basically, in each and every organic molecule, the things that we want to remember is the chemical basics of it. What are the elements of each and every molecule? So up to now, we complete the carbohydrate. It is carbon, hydrogen, and oxygen elements. What are the main elements? Carbon, hydrogen, and oxygen. And when we come to the starting unit or the basics unit of carbohydrate, it is monosaccharides. And when we go through the classification of carbohydrates, they are monosaccharide, disaccharides, and polysaccharides. Monosaccharides, disaccharides, and polysaccharide, and you learn what are the examples for them also. So from this onwards, I will give you only the most important thing, only the important factors that they will ask in the exam. So don't worry, so it means when we go through the past papers and when we analyze the past papers, most of the times they are focusing specific area. So when we are targeting our exam, better to go through with that. So if we are able to write a long note, it is not need. Okay, just the most important factors into one note, keep that in your note and study it well. Then you can easily go through and understand what are the mistakes, and you can easily, when you go through the past papers, you can easily find marks. Okay, gain some marks from past papers also. Yeah, so let's see, write significance of carbohydrate. First, let's see read. Then after I will explain. Just quickly read it. There are 4 significations. Can you quickly read it?
As an energy source,
Yeah.
the main source to obtain energy for the activities of organisms is the carbohydrate. The monosaccharide, its name glucose, produced due to hydrolysis of those compounds, release energy.
Okay.
As a storage compound, as a structural component in plant cell wall, as a constituent of nucleic acid.
Yeah, so when we go through this one by one, first one is as an energy source. We all know we eat this carbohydrate food just like rice, potato. Okay, basically those things give us some sort of energy. Okay, so the basic requirements of this carbohydrate is gaining energy for us. So that is the basic thing. Okay, basic significance. Then we come to the second one, as a storage compound. You already know when you go through the polysaccharides, the starch is the substance then storage in plants. When we come to the animal, glycogen is the substance that storage in animal tissues and muscles. Then when we come to the next point, the structural component of plant cell wall. Can you remember the structural component of plant cell wall? It is a polysaccharide. It is cellulose. It is cellulose. Other than that final point, as a constituent in nucleic acid, I will deeply explain this in the point that we are learning about nucleic acids. I think it's all clear. Let's see, write down the row. So when you are writing the note, try to put that into your membrane. It means try to put that in your brain. Try to study while you are writing the note, because we don't have another time for studying. We have to, within the time that we are learning right now, try to keep them in your mind also. Let's see, write down the first significance is, write down the first one, as an energy source. As an energy source. As an energy source. As an energy source. Second one, as a storage compound. As a storage compound. As a storage compound. First one as energy source. No need to explain, just the points. First one as energy source. Second one as a storage compound. As a storage compound. As a storage.
Okay, sir.
Third one as a structural component in cell wall. 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 Final one, as a constituent of nucleic acid. As a constituent of nucleic acid. As a constituent of nucleic acid. Okay. So then after Daruo, we are learning test to identify carbohydrate. Basically, this is a very important one because in the structured paper, mainly they will focus on this. Okay. Write down test to identify carbohydrates. Test to identify carbohydrates. Test to identify carbohydrates. Write down the first one, Dua. When we go through the carbohydrate, there are mainly three parts. They are monosaccharides, disaccharides, and polysaccharides. Here, we are doing three tests to identify each: polysaccharides, monosaccharides, and disaccharide. Let's go with the polysaccharide first. Write down starch test. The main topic is test to identify carbohydrate. The subtopic is starch. Starch test. Starch test. Starch test. Starch test. Yes. Let's go with the next point. Let's go with the explanation. This is the easiest test that you are doing. First of all, you have to do, you have to take a solution of starch. So here, they have mentioned small amount of food is obtained and grind well with water. It means that we have to take a small amount of carbohydrate food. It can be potato, it can be rice, so it depends. Okay, it can be sugar. Different types of materials include carbohydrates as well as starch. So you can take any sort of bread also. Okay, you can take a bread part and grind it and prepare a solution. We need something include starch. Okay, take the food sample, then you have to put a drop. After preparing that solution, a drop of iodine is added to the above solution. Only the thing that you want to do, initially prepare the solution. Second point, or then after you have to put the iodine solution to that. Then you can easily see the color change. So normally iodine, the color is brown in color. So that brown color converts to purplish blue color. Then you can actually identify it is starch. Starch is present in that food. All clear? Good. Do I want to explain it again? Are you clear with it?
Can you please, sir?
Yeah, okay. Daro, here, this is the thing. Okay. So if you, this practical is done to identify starch. Okay. First of all, you have to take the food sample. It can be a food that includes starch, or if it is okay, if it does not include starch also. First of all, take the food sample and prepare a solution. It means we have to grind it and mix with water, and we have to prepare a solution. Then after, the thing that you want to do, take the iodine solution and put few drops to that prepared solution. If this prepared solution includes starch, definitely it will give the color change. If not, if this prepared solution does not include starch, there is no any color change. What is the color change? Initially iodine is brown color, then after that color change to purplish blue color. If the color change from brown to purplish blue, we can identify this food includes starch. If not, if there is no any color change that you can observe, then you can say this food does not include starch. Is it clear? Do I want to show you a video clip to clarify it? I think it's okay with you.
No, sir. It's clear.
Yeah. Okay. Let's go with the video then. Yeah. Can you see the screen?
Yeah, sir.
Yeah. Yeah, no? Okay. Starch identification test. Yeah, this is the one. Okay. Let's go through with this. When you watch it, you can clearly understand.
An important constituent of our daily food are carbohydrates, which we consume in the form of starch. Starch is a high calorie food that converts to sugar during digestion. Starch is also produced by all green plants as an energy store. Today, we will conduct an experiment to test the presence of starch in potato, rice, sugar, and salt. For this, we will need iodine solution, which is yellow brown in color. Note the change in the color of iodine as it is put on the rice and potato. Both turn the iodine blue-black. Put some iodine on the sugar cubes. The color does not change. Salt too does not change its color. This proves that sugar and salt do not contain starch, while the starch in rice and potato reacts with iodine to form a blue-black compound. Now the reaction is because the blue-black color is basically the formation of polyiodide chains from the reaction of starch and iodine.
Yes. Yes, Daru. Is it okay with you? Now clear? The only thing, you have to put a few drops of iodine. If there is a color change in iodine from brown color to purple color, it means this food includes starch. If there is no any color change, it means this food does not include starch. All clear? Got it?
Yes, sir. Got it.
Good. Okay, let's go with the second experiment. Write down test for glucose. Did we write the test for the starch? Not yet, no? Did we write the note for that?
No, sir.
No. Okay. Yeah, we have to write it. First one, write down starch test. Write the steps, only two steps that you want to follow. Write down starch test. First one, small amount of food is, a small amount of food is, small amount of food is obtained and obtained and obtained and grinded well with water, grinded well with water. Small amount of food is obtained and grinded well with water, grinded well with water. Second point, second step, a drop of iodine solution is added, a drop of iodine solution is added to the above solution. A drop of iodine solution is added to the above solution, to the above solution. Then write the color change the rule, write the observation, write a point, observations and write observations, observations. Purplish blue color appears, purplish blue color appears, purplish blue color appears. If it appears with purplish blue, we can confirm it includes starch. If not, we can confirm there is no starch in this food. That is the experiment that you want to go through, you want to understand. That is it. Okay. Then let's go through with the next point here. Right, number 2, second one, test for glucose, test for glucose, test for glucose. Second experiment. Now we identify first, from the first experiment we go through with starch, it is a polysaccharide. From this we have to go through with Glucose, it is a monosaccharide. Okay, let's see, read the steps. Can you read the steps that you want to follow to identify glucose? Just read it.
A solution of glucose is obtained into a test tube. Few drops of Benedict solution to the above solution is added.
Okay.
The above solution is immersed in a water bath and heated.
Okay.
Then observe color changes as below.
Okay. Taro, you have to remember this color difference. Okay. Definitely, they will ask this color frequency. You have to keep this in your mind. First color. So if the sample include glucose, it gives this color change. Okay. If the sample does not include glucose, there is no any color change, only the initial color. So here, what they have done, a solution of glucose is obtained into a test tube. So we all know that there are a lot of ways that we can take glucose. So in the packet also commercial you can find glucose. So then you have to dissolve it in water and prepare a glucose solution. Then we have to put it to a test tube. Then after that, the thing that you want to do, put few drops of Benedict solution. Benedict solution also we can find in our labs. It is just named with the Benedict. So we can take the solution out from that and we can add few drops of Benedict solution into the test tube. Then the above solution is immersed in a water bath. It means we have to heat it. So first of all, we have to immerse it in water bath and heat. While the process going through there, we can see this color change. Keep that in mind. What are the colors? Blue color, green, green yellow, brick red. Blue color, green, green yellow, orange, brick red. To identify this clearly, definitely you have to go through with the video. Okay, so I will do like that. So give me 1 second, I will share my screen. Let's see, go through the video to identify is this, because this is a bit complex experiment than the previous one. Here initially we went through with the starch identification test. Now we are going through monosaccharide. It means glucose identification test. Okay, so the Benedict solution is the one that we are using here. So this shows you the color change. Basically understand what will happen there.
We are going to perform the Benedict test. The apparatus used for this test are test tubes, test tube stand, a test tube holder, measuring cylinder, and a beaker containing the sample. The reagent used for this test is Benedict's reagent. Take a test tube and hold it firmly with a test tube holder. Add 5 ml of Benedict's reagent. Add 8 drops of the sample. Shake the contents well and boil it for approximately 1 minute. Shake the test tube by heating it. There is a formation of a color change which indicates the positive Benedict test.
Yes, that's it. So when we do those experiment in practical scenario, we can't observe this much of clear color difference. Okay, but in the notebook they have given you specifically. So in our labs we don't have that much of facility. So normally we can't see that much of clear color change. But no, the thing that you want to understand, if they ask the question in the exam, you have to write this color sequence. What are the color change when a glucose put into a Benedict solution and heat? If they ask it like that, you have to give this color sequence. Is it clear? Do I want to explain it again? All good?
No, sir.
Clear, no?
Yes, it's clear.
Yeah, very good. Nice. Let's see, write down the row. Write down. Test for glucose. Test for glucose. Write down the first one. A solution of glucose is obtained into a test tube. A solution of glucose is obtained into a test tube. Then few drops of Benedict solution. Few drops of Benedict solution. Few drops of Benedict solution. Few drops of Benedict solution added to the above solution. Few drops of Benedict solution added to the above solution. Second point. Few drops of Benedict solution added to the above solution. Third point. The above solution is immersed in water bath and heated. The above solution is immersed in a water bath and heated. Immersed in a water bath and heated. Immersed in a water bath and heated. Final point. Observe the color change. Observe the color change. So, you have to write this color sequence and highlight it, or use a different pen, red pen or something else to write down this, because you have to highlight, you have to emphasize this, and you have to remember this way. First one, blue. Second one, green. Blue, green. Third one, green, yellow. Green, yellow. Fourth one, orange. First one, blue, then green. Third one, green, yellow. Fourth one, orange. Final one, brick red. It is not only a color, okay? It is a precipitate. Write down. Brick red precipitate. Brick red precipitate. Brick red precipitate. That's it. Brick red precipitate. Okay, Daro, let's go with the experiment number 3. Test for sucrose. Test for sucrose. Just as the previous one, basic steps are same, but finally we are doing a different one. Just read that also.
A sucrose solution is obtained into a test tube. Few drops of Benedict solution is added to it. The test tube is immersed in a water bath and heated. No color change. Few drops of diluted sulfuric acid is added to the freshly prepared sugar solution and heated.
Okay.
Next, few drops of Benedict solution is added to it. Can
Okay.
observe color changes as below.
Yeah, it is also the same. The color change also same. But only thing, when we are doing the experiment for sucrose, first of all, we have to do the basic steps that we have done in the previous one. Take the sucrose solution and few drops of Benedict solution, and we have to heat it. But as the initial step, it does not give any color. That observe, it does not include glucose. In the second step, we have to take, this is the different step. Then we have to use dilute sulfuric acid, freshly prepared dilute sulfuric acid. We have to put it and heat it. Then after again the previous one, Benedict solution, and while heating in the Benedict solution, we can see this color change. Only the difference is we have to use dilute sulfuric acid and freshly prepared sugars. We have to take dilute sulfuric acid and put to the freshly prepared sugar solution. Okay, let's see, write down the rule. A sucrose solution is obtained into a test tube. A sucrose solution is obtained into a test tube. This is the test for sucrose. Okay, first of all, you have to write the topic, test for sucrose. Then after write, a sucrose solution is obtained into a test tube. Second one, few drops of Benedict solution is added to it. Few drops of Benedict solution is added to it. Few drops of Benedict solution is added to it. Few drops of Benedict solution is added to it. Third one, the test tube is immersed in a water bath and heated. Just like the previous one. The test tube is immersed in a water bath and heated. If it includes glucose, it will give the color change at this point. But here there is no any glucose, then there is no any color change. Write down, the test tube is immersed in a water bath and heated. Next point, no color change. There is no color change because this solution does not include glucose. No color change. No color change. Then few drops of diluted sulfuric acid. Few drops of diluted sulfuric acid. Within the brackets, write down the row H2SO4. Few drops of diluted sulfuric acid. Within the brackets, write down H2SO4 is added to a freshly prepared sugar solution and heated. Is added to a freshly prepared sugar solution and heated. Sugar solution and heated. Sugar solution and heated. Then write the next point. Next few drops of Benedict solution is added to it. Next, few drops of Benedict solution is added to it. Finally, the row, write down. Can observe the color change. Can observe the color change. Can observe the color change. Write down the colors. Blue. Can observe the color change. Write down the colors. First one, blue. Blue color, blue. Then green. Blue. Green. Green, yellow. Orange. Recreate precipitate. Those are the points. Blue, green, green, yellow, orange. Recreate precipitate. Recreate precipitate. Okay, that's it. Yeah, so from that we can conclude with the carbohydrates. Let's go with the next point. Next topic, it is proteins. Write down to proteins. Can you read it? About proteins.
Protein is an essential constituent in all living cells. Carbon, hydrogen, oxygen, and nitrogen are always present in proteins. Sometimes sulfur can also be present. 17% of the major human body is composed of proteins. Protein is a complex molecule made up of polymers
Polymerized. Okay, good. Polymerized.
Polymerized acid molecules meat, fish, egg, white, so that come
Yeah. Egg white, cereals are some of the foods rich with proteins. Okay, that I will explain this. Protein is an essential constituent in all living cells. You already know that. Okay. So when we go through the elements of protein, Darwo, you have to remember there are 5 elements, but 4 elements are essential. It means it definitely each and every protein include this carbon, hydrogen, oxygen, and nitrogen, sometimes sulfur. So sulfur, we can't find sulfur in all the amino acid, but some sulfur is present, some are not. Then we come to the next point. 70% of mature human body is composed of protein. It means that when we consider, just think that my weight is 100 kilograms. So from 100 kilograms, 17 kilograms is from protein. All the other nutrients have 83 kilograms, just like that. Protein is a complex molecule made up of polymerized amino acid. It means the basic unit of protein is amino acid. Finally, the foods that contain protein are normally the fish and meat-based products include high protein than vegetables. So if you are vegetarian, definitely you have to go through with some sort of protein supplements, and we have to take some extra protein for our maintenance of body. Is it clear? Do you have any question? I heard that you're asking something from me. Anything to clarify?
so amino acid molecules mean
Yes, Daru. Amino acid means the basic unit of protein. Just like monosaccharides, when we learn carbohydrate, the basic unit of carbohydrate is monosaccharide. It means starting unit or the smallest unit. In protein also, the smallest unit is monosaccharide. In extra reading part, though, they have given the structure of monosaccharide here. They won't ask questions on it. But if you request, I can teach this also. But first of all, let's write the important points from this. Then after, I will explain you what is the amino acid and how the structure forms. Okay. Yeah, quickly write down. Main elements of protein is main elements of protein is main elements of protein write down the rule. Write down first one carbon, then hydrogen, then oxygen, nitrogen. Carbon, hydrogen, oxygen, nitrogen. Carbon, hydrogen, oxygen, nitrogen. Carbon, hydrogen, oxygen, nitrogen. Write down always present in proteins. Always present in proteins. Always present in proteins. Next point. Sometimes sulfur can also be present. Sometimes sulfur can also be present. Sulfur can also be present. Sulfur can also be present. Sometimes sulfur can also be present. Next point, Daru. Next point. Next point. The basic unit of proteins are amino acids. The basic unit of protein, amino acid. Amino acid. It is amino acid. Then write down the foods that rich with protein are the foods that rich with proteins are the foods that rich with proteins are the foods that rich with proteins are write down the row examples: meat, fish, egg white, cereals. Meat, fish, egg white, cereal. Meat, fish, egg white, cereals. That's it. That are the points that you want to remember. So as you ask in the question on amino acid duo, this is the structure of amino acid they have given you. So normally no need to write these things. Okay, these are extra reading part. I will just explain this. Basically, when you come to the amino acid, there are 4 parts. One part is amino group, another part carboxyl group, and H as well as R. So from protein to protein, it means amino acids to amino acid, this R part change or varies. So in here, R represent the group containing both carbon and hydrogen. Due to the change in R group, 20 different amino acids are present when you go through this. Normally, there are 20 different amino acids. All proteins present in organisms from bacteria to human are made from these 20 different amino acids. As I explained you, the basic unit of protein is amino acids. So there are 20 different types of amino acids. These are the ones who help to prepare protein. A simplest amino acid is glycine. It means this R part is the part that have the ability to vary. But when it replaces hydrogen, we call it as glycine. Here the H is present as in the R group. So those are the things you want to. No need to remember. Okay, it is extra knowledge one. Other than that, they have given you some of the examples. It is also extra knowledge. Examples that we can see in our day-to-day life. Basically, protein present in our muscles, we name them as actin and myosin. Protein present in our bones is osein. Protein present in our blood cells, we all know that is hemoglobin. Present in hairs, it is keratin. Present in leguminous food, it is legumin. Present in wheat flour, we already know it is gluten. Finally, proteins present in egg white, we call albumin. Okay, that's it. All those things are extra reading part. And here, definitely this part you have to remember very well. It is about the significance of protein. Significance of protein. But it is easy compared to the other substances. These are very practical things that we know, that we learn in our day-to-day life. Let's go through it. Just read it, the points.
As an energy source,
Okay.
when energy supply from lipids and carbohydrates is not sufficient, protein is used in energy generation
Very
to
good.
make structural components. Proteins are important components in making cell membrane.
Okay.
Hairs and feathers also contain keratin protein.
Okay.
As enzymes,
Science.
all the biochemical reactions take place in organisms are catalyzed by enzymes. The enzymes are proteins.
Okay.
As hormones,
Mhm.
some hormones are proteins which involve
Homeopathy. Yeah.
homeostasis and
Coordination.
coordination of organisms.
Very good.
As antibodies,
Mhm.
the antibodies that are produced in the body to protect the body against microorganisms that enter into the body are proteins.
Yeah. So when we go through the importance or the significance of proteins, as we already know, carbohydrate is energy source, protein is energy source, lipid also energy source. So it is an energy source. First point is clear, very clear with you. Second one, to make the structural components, you already know, in our cell membrane, in our hair, in each and everything, it includes protein. So normally it is a component or structural component that we see, that we can see in our day-to-day life. Third one, as enzymes, we will learn this later. In rate of reaction chapter, we will deeply learn these enzymes. We call them as biochemicals or biological accelerators, because they accelerate the rate of reactions that take place in our body. So the enzyme also a part of protein. It means a type of protein. Hormones also are not like enzymes. The hormones transport through blood and do different type of tasks. And it basically helps to the coordination or maintain in our body functions very well and prepare a good relationship between it. Final one, antibodies. In nowadays we are deeply learning of those things. We are deeply we having discussions of those 19. You already know in COVID-19 what happened. The immunity, for the improvement of immunity, to improvement of ability to fight with that bad virus, we are taking the doses of vaccination, first dose, second dose, and booster dose. So from those, we are artificially taking those antibiotics for our body. Rather than that, in our body, we have different functions to produce those antibodies as well. All clear? Good with you? Okay, just,
Yes, sir.
yeah, okay. Let's write down the points. Write down. First one, the main topic is significance of protein. First point, as the energy source. As the energy source. You have to remember those points on as energy source. Second one, to make structural components. To make structural components. To make structural components. To make structural components. As enzymes. As enzymes. As enzymes. As hormones. Third one as enzymes. Fourth one as hormones. As hormones. As hormones. As hormones. As hormones. Final one as antibiotics. As antibiotics. As antibiotics. As antibiotics, or we can call it as antibodies. Antibodies. As antibodies. As antibodies. As antibodies. As antibodies. Yeah, so in here, then after we are focusing on test to identify protein. Just read it though. We have to go through with the video clip to just to clarify and understand the process. First of all, let's read it. Test to identify proteins. It is biuret test. Biuret test. It is also easy one. Let's go through. Just read it. Just read it.
A solution made by grinding dye and egg white is obtained into a test tube. An extra amount of sodium hydroxide and then few drops of copper sulfate is added into it. Solution turn to purple color.
That is the observation. Okay. So let's try to understand. Let's try to go through with this test. If try our best to find one. Note that. Yeah, this is the biuret test. Yeah, this also, yeah, it is better.
Egg white contains a lot of protein. The test for protein is called the Biuret test. This involves adding some sodium hydroxide solution, then running some very dilute copper sulfate solution down the side of the tube. At the top of the mixture, the colour change to mauve shows that protein is present. So which of these will test positive for protein? Bread in tube 1, nuts in 2, and grape juice in 3. Both the bread and nuts contain protein. Which contains more?
Yeah. Okay, that is basic. So the only thing that you want to do, first of all, you have to prepare a solution. Prepare a solution based on protein. Then after, what do you want to do? Then after, you have to put sodium hydroxide and a few drops of copper sulfate to the solution. If they give you a color change, it means it includes proteins. If they does not give you a color change, it means they have the ability, but they don't include any proteins in it. Write the topic. Test to identify proteins. Test to identify proteins. Test to identify proteins. Write the first one, biurate test. Biurate test. Biurate test. Then write the points. Write down biurate test. A solution made by grinding dal or egg white. A solution made by grinding dal or grinding dal or egg white is obtained into a test tube. Obtained into a test tube. Then an extra amount of sodium hydroxide, within the brackets write down NaOH. An extra amount of sodium hydroxide, within the bracket write down sodium hydroxide, NaOH, and few drops of copper sulfate, is added into it. Within the brackets write down the formula for copper sulfate. It is CuSO4. Copper sulfate is added into it. Then write down the next point. Solution turns to purple color. The solution turns to purple color. It gives a positive value. The solution turns to a purple color. That is the observation that you can see. The solution turns to a purple color. So then after we are coming to the next one, Daru, it is also a part of protein, and we call it as enzymes. But as I told you previously, we will learn on enzymes, their catalyst, and how they work as a catalyst and in next stages. But in here, they have given you the basic idea for the enzymes and what are the importance and how do you going to clarify enzymes based on other properties. Write the topic enzymes. Enzymes. Okay, enzymes. Write the topic enzymes. Yes. Daru, I think when we go through this one, better to read this also. Yeah, just read this one.
Special proteins that are produced within the organism to increase the rate of biochemical reactions are known as enzymes.
Okay.
For example, to convert sucrose into glucose, sucrose has to be heated with a dilute exit, but the enzymes present in the digestive system do the same reaction at a low temperature.
Okay.
Therefore, the activity of enzyme is to catalyze the biochemical reactions.
Okay.
Engaging the following activity on how enzymes function.
Yeah, so it is activity that we can go through to identify the functions of enzyme, how they increase the rate of reaction. Though, first of all, let's see, write down the special points on this. Okay, write down. After writing enzyme, write down a special proteins within the brackets, write down organic catalysts, special proteins within the brackets, write down organic catalysts that are produced within the organism, to increase the rate of biochemical reactions are known as enzymes. Write down the next point. Write down the activity of enzyme is to catalyze the biochemical reaction. Catalyze the biochemical reactions. Okay. They will accelerate the reaction rate of biochemical reaction. Catalyze the biochemical reaction. That's it. Okay, they have given you an experiment for this. Write the topic. Activity of amylase on starch. This is a very easy one. No need to write. Okay, I will just explain this. Then you can easily understand how and what the process going on. First of all, you have to read it. Let's see, read it first.
Material required: flour, amylase, test tube, white porcelain tile, iodine solution, water, a stopwatch. Method: put 2 ml of starch solution into a test tube. Add 2 ml of amylase into it and mix well. Get a drop from the solution after 2 minutes and place it on a white porcelain tile. Add a drop of iodine onto the drop of mixture. Continue same procedure for about 20 minutes in 2 minutes intervals.
20 minutes in 2 minutes interval. Okay, that was. So in here, so I will request you to write these materials and methods as a homework, because in here now I will explain it to you. Okay, don't waste your time on writing those things. So I will explain this and clarify it to you. Basically, write the first subtopic, material required, then write method, keep some space for that, and now listen to it carefully. Okay, here what they have done, as the materials they have used flour, amylase, test tube, white porcelain tile, iodine solution, water, and stopwatch. These are the things that they have used. So what the thing they have done in here, so this amylase, it is an enzyme. It has the ability to break down the starch. Okay, so that's why they are taking 2 milliliters of starch solution in the test tube. Again, add 2 milliliters of amylase. What will happen? Amylase convert that starch to monosaccharides. It means basically the basic monosaccharides. It has the ability to convert it into monosaccharides, basically maltose, and mix well. get a drop from the solution. We have taken the drop from the solution that we already put starch and amylase, and we are doing the test. Get a drop from the solution after 2 minutes and place it on a vial porcelain tile. Add a drop of iodine into the drop of mixture. Continue the procedure to 20 minutes from each 2 minutes interval. What will happen, Daruo? So initially it includes starch. So after adding amylase, what will happen? That starch may convert to maltose. So when we do this iodine test give the positivity only for the starch. So while we are taking the observation every 2 minutes for 20 minutes, when we come to the stage from 15, 16 minutes, almost all the starch in the solution may react with the opponent. Okay. All the starch, basically almost all the starch in the component may react. Okay. That is the basic thing. Almost all the starch may react with it. But after this amylose enzyme has the ability to remove all the starch in the solution. After that, when we put iodine drop into the solution, we can't see the color change. It means it has the impact on color change also. Yeah, it is the basic thing and it has the impact, but we are not focusing that much. Okay. So until almost all the starch is converted to maltose, this iodine give the positive mark. It means it will change the color. But after removing all the glucose, it means starch into maltose, there won't be any color change. That is the basic practical that we can do to identify the effect or the properties or the functions of proteins. You have a homework, you have to write the materials and methods in the given space. And now you have another task. It is to read the note. Yeah, quickly read it.
The blue color of the drop obtained from the mixture gradually reduces with time and finally obtains the color of iodine,
Okay.
yellow or brown color.
Okay.
Starch gives black-blue color with iodine, but it does not give color change with iodine after 20 minutes as there is no starch there.
Mhm.
That is because starch is converted to maltose by amylase enzyme.
Enzyme. Yeah, that is the thing that I already explained. You write down the row, write down. Okay, you have to keep the space for the procedure also. Method, then write down. The blue color of the drop obtain the blue color of the drop obtained from the mixture gradually reduce with the time gradually reduce with the time or reduces with the time gradually reduces with the time and gradually reduces with the time and finally obtains the color of iodine. Finally obtains the color of iodine. Within the brackets, write down the row color of iodine. Finally obtains the color of iodine. Within the brackets, write down it is yellow or brown color. Yellow, it can be yellow. Normally it is brown. Yellow or brown color. Then the next point, write down. Starch gives blue color with iodine. Blue color with iodine. Starch gives blue color with iodine. black, blue color with iodine, but it does not give color change. But it does not give color change. But it does not give color change with iodine. Color change with iodine after 20 minutes. 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 As there is no starch there. 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 That is because the starch is converted to, that is because the starch is converted to, that is because the starch is converted to, the starch is converted to, the starch is converted to maltose by amylase enzyme. The starch is converted to maltose by amylase enzyme. Maltose by amylase enzyme. Maltose by amylase enzyme. Maltose by amylase enzyme. So that's it. That is the purpose of enzymes. And we will deeply learn those things at the end of our chapters, end of our grade 10. Yeah, grade 10 part on enzymes are the factors that important for the rate of reaction. Okay, Daru. So let's see, we can come for the third one. So we already complete carbohydrates. We already complete proteins. Now we are coming up with lipids. Write the topic, lipids. Write the topic, lipids. Lipids. Yeah. First of all, let's read it.
Fats and oils belong to this group. Lipids which are solid at room temperature are called fats and lipids are called oils. Similar to carbohydrates, fats also contain carbon, hydrogen and oxygen as constituent elements, but a lipid contains much less oxygen than a carbohydrate. Lipids are insoluble in polar solvents including water. They are soluble in organic solvents. Groundnut, coconut,
Gingerly.
gingerly, butter and margarine are examples for foods which contain lipids. Fatty acids and glycerol react to form lipids.
Okay, good. So here, now we have to focus on lipids, Daru. Basically, when we compare with the carbohydrates and proteins, these lipids are a bit complex. Okay, the basic and main point that you want to understand, Daru, there are two types of lipids. We call them as fats and oils. Fats and oils. Normally, in our room temperature, fat can be obtained in solid form, but when you come to the oils, it is in liquid form. Fat, what is the type of fat you can see in our day-to-day life? It is butter, no? Butter is in solid form. What are the oils that you can see? Sunflower oil, vegetable oil, corn oil. So there are different types of oils. Okay, sunflower, okay, olive oil, those things are in liquid form. So the ones we can find as once we can find in solid form, we call them as fats. Once we can find in liquid form, we call them as oils. So similar to the carbohydrate, there are chemical basics or the components or the elements of lipids are carbon, hydrogen, oxygen, and there is a difference. You have to remember that also. When we compare with the carbohydrates, lipids include less oxygen. This was questioned in past paper. What is the difference between carbohydrate and lipid in their chemical structure? You can say the basic unit of carbohydrate are monosaccharides. The basic unit of lipids, there are two basic units here you have mentioned, fatty acid and glycerol. Other than that, the elements are same, okay? Carbon, hydrogen, oxygen, same for both carbohydrates and lipids, but lipids include less oxygen than carbohydrates. Second, next one, lipids normally insoluble in polar solvents because there is a theory that we are learning. Normally, polar solvents, the dissolving ability taken from polar solutes. Normally, polar solutes will dissolve in polar solvents. So we know lipid is non-polar. So we can't dissolve non-polar lipid in polar solvent like water. So if we want to dissolve lipid, we have to take a non-polar solvent for that. Just like different types of, there are different types of non-polar liquid. Kerosene we can use. Petrol, diesel, those are organic solvents. And not only that, acetone, also different types of organic solvents that we can use to dissolve lipids. Definitely they should be organic as well as non-polar. So they have given you the examples also, groundnut, coconut nut, ginger, butter, and margarine are the examples. And here, this is the way that they form. Fatty acid plus glycerol produce lipids plus water. So when we go through the basic unit of lipids, there are two basic units, not only one. For the carbohydrate, it is only monosaccharides. For the proteins, it is only amino acids. But when you come to the lipid, there are two types. They are fatty acids and glycerols. Okay, let's write down. Write down. When we focus on the physical properties, lipids can be divided into two categories. When we focus on physical properties, lipids can be divided into two categories. Lipid 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 can be divided into two categories. They are fats and oils. They are fats and oils. They are fats and oils. They are, don't write them in paragraph, just write them in point wise. They are fats and oils. Next point, write down the rule. Next point. Lipids which are solid at room temperature. Lipids which are solid at room temperature. Solid at room temperature. Solid at room temperature. Solid at room temperature. And lipids which are, write down. Lipids which are solid in room temperature are called fats. Are called fats. And are called fats. Name. Fats. And liquids are called as oils. And liquid called as oils. And liquids called as oils. And liquids called as oils. Liquids called as oils. Then write down main elements of lipids are main elements of lipids are carbon, hydrogen, and oxygen. That is it. The main elements of lipids are carbon, hydrogen, and oxygen. Carbon, hydrogen, and oxygen. Write down the next point, Daru. Next point. But lipids contain much less oxygen than carbohydrate. But lipids contain much less oxygen than carbohydrate. It has low amount. Okay? But lipids contain much less oxygen than carbohydrate. Much less oxygen than carbohydrates. Much less than carbohydrate. Next point, write down the row. Next point. Lipids are insoluble in polar solvents like water. Lipids are insoluble in polar solvents like water. Lipids are insoluble in polar solvents like water. Water. They are soluble in organic solvents. 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 Then write down the next points, Dua. Write down next point. Examples. Just write the examples. E.g. examples. E.g. examples. E.g. examples. Write down. Groundnut. Groundnut. Coconut. Groundnut. Coconut. Gingerly. 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 Butter and margarine. Butter and margarine. Gingerly. Butter and margarine. 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 Okay, then write this formula also. Fatty acids plus glycerol converts to lipids plus water. Fatty acids plus glycerol convert to lipids plus water. Lipid and water. Fatty acids plus glycerol convert to lipid plus water. Lipid plus water. Lipid plus water. Okay, let's go through with the significance of lipids. Significance of lipids. Let's go through significance of lipids. Let's read it through.
As an energy source, lipids act as an energy source as carbohydrates and proteins. More energy is produced during burning of lipids. To form different structural components, lipid is one of the most important compounds in cell membrane, especially phospholipids, and cholesterol. For,
Cholesterol. Okay.
cholesterol. For conservation of water,
Okay.
the wax known as protein present on the surface of the plant body conserve water. Most animals' protein converting also contains wax, which helps to avoid digestion as it is impermeable to water. To maintain the body temperature, warm-blooded animals such as birds and mammals possess a hypothermal fat layer which acts as a thermal insulator.
Okay.
It helps to maintain their body temperature. Fourth one, to protect internal body organs. The fat layer surrounds the organs and structures in the body and absorbs external shocks, thereby provides protection. Fifth one is to synthesize some hormones. Some hormones of vertebrates are
Estrogen,
estrogen,
testosterone, and cortisol.
testosterone are lipid compounds.
Yeah, lipid compound. So here when we compare with the protein and carbohydrate, the lipid has lot significance. Okay, so it means importance. Let's see, go through one by one. The thing that you want to remember, only the points Daru. Basically, when we go through this significance, first one is an energy source. We all know lipid is an energy source. Up to now, carbohydrate, proteins and lipid, all the three are important energy source. But you have to remember the sequence also. When we go through the sequence or the way or the remedy that we are consuming those energy sources, basically first of all we have to take the energy from carbohydrates. So after consuming all the carbohydrate in our body, if we need more energy, we have to move forward with lipids. If we take use of all the carbohydrates as well as all the lipids, then after we have to move forward with the next option, it is the proteins. So it is a basic thing. Normal carbohydrate, protein and lipid, all the three sources are energy sources. Second one, that was to form different structural components in carbohydrate. Also it helps to form cell walls. Protein also our hair and different parts. In that sense, this lipid also very important as a structural component basically in our cell membrane, especially for the lipids and cholesterols. Sorry, phospholipids and cholesterol are the type of lipids that can find in cell membranes. For conservation of water, basically almost all the species, basically when we come to the plants, they have a cutin layer. In each and every plant leaf, there is a protection layer named as cutin. It prevents the evaporation of or removal of water from the plants. So when we come to our body also have a fat layer. From that, it reduces the evaporation of water from our body. That is the one they have explained with the conservation of water. Then the next one, to maintain the body temperature, not only the conservation of water, but also to maintain a constant body temperature. It is important for us because there is basically for the mammalians, when we go through the classifications of animals, there are ones with, when we come to the vertebrates, basically fishes, amphibians and reptilians, their body temperature fluctuates with the, basically they have different type of body shapes, but their body temperature fluctuates with the environmental temperature. But when we come to the fishes and mammalians, it means birds and the ones who depend on their mother's milk, it means mammalians, their does not fluctuate their temperature according to the environmental temperature. They have the ability to maintain specific temperature within their body. For that, this fat layer is very important, because from this fat layer, we are able to maintain the temperature that we have in our body. Yeah, that's why they have explained the warm-blooded. Okay, basically, as I explained you, the fishes, amphibian, reptilians, those are cold-blooded animals. It means their body temperature fluctuate with the environmental temperature. But when you come to the warm-blooded animals, just like fishes and mammalians, fishes aves and mammalians. Aves means birds, mammalians means the one who depends on their mother's milk. They have the ability, basically they have the properties, basically ability. What is the purpose of that? They can maintain, because of this hypodermal fat layer. We have a fat layer inside our skin. There is a fat layer. Because of that, we can maintain our body temperature. Next one, protect the internal body organs. Normally, we are doing different types of exercises, different types of sports, cricket, rugby, and anything else. So in those sports, we will do, and we have different damages, injuries, each and every part of our body. But there is no effect for the internal organs. Okay, what are the internal organs? Our lungs. Okay, basically our heart, our digestive system, our kidneys. Almost all these internal organs are covered with the fat layer. It is just like a rubber. So when we hit to a rubber, there is no any harmful, because it have the ability for stretch and overcome those pressures that we are providing. So almost all the internal organs are covered with this fat layer. Because of this cover, we can easily overcome this problem. Then after that, to synthesize some hormones of vertebrates. Basically, as I explained you, there are some hormones that produced by proteins also. But not only proteins, but also lipids also have the ability to produce different types of hormones in vertebrates. They are estrogen, testosterone, and cortisol. Those are the ones that we can see. Yeah, let's see, write down that, Darvo. Let's see, write down significance of lipids. Write the first one as an energy source. As an energy source. Only, just write the points. Okay, no need to explain. As an energy source. Write the first subtopic, significance of lipids. First one, as an energy source. As an energy source. As an energy source. Second one, to form different structural components. To form different structural components. To form different structural components. Different structural components. Third one, for conservation of water. 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 Next one. Next one. This one. To maintain the body temperature. 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 Maintain the body temperature. Next one, to protect internal body organs. To protect internal body organs. To protect internal body organs. Internal body organs. Internal body organs. Internal body organs. Final one, Daru, write down. Synthesize some hormones. Synthesize some hormones. It will help to synthesize some hormones. Synthesize some hormones. Synthesize some hormones. Okay. Synthesize some hormones. Yes. Yeah, with that, we are coming for the final part of the lipids. Write down. Test to identify lipids. Test to identify lipids. Test to identify lipids. Write down the name of the test is Sudan 3 test. 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 Let's read that first.
Some amount of gingerly oil or coconut oil is added into a test tube. Student reagent is added into it. Appearance of red fat global
Globules. Yeah, that's it. That is the easy one, Daru. We just want to take a fat sample. It can be fat oil, they say gingerly oil or coconut oil. Then after the indicator we are using here, it is Sudan 3. After putting Sudan 3, it should be appearance. We have to, we should have the, we have the ability to see red flat, red sometimes it may be pink also. Red fat, basically write this because they have given it red fat globules. That is the easy one only. Easy observation, just like starch identification, just put in iodine and see the color change from the purple to dark colors. So in here also, just take a solution of fat or oil, put few drops of Sudan 3, then you can see fat, red fat globules. Let's write down. So, some amount of gingerly oil or coconut oil, some amount of gingerly oil or coconut oil is added into a coconut oil is added into a coconut oil is added into a test tube added into a test tube Next one, Daru, write down. Sudan 3 reagent is added into it. Sudan 3 reagent is added into it. Sudan 3 reagent is added into it. Added into it. Added into it. Added into it. Finally, write down the observations to it. Observations. Appearance of red fat globules. 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 You can see red fat globules. Red fat globules. appearance of red fat globules. That's it. That is the end of fat, and we are coming for the next one. Write down nucleic acids. Nucleic acids. That is the easy one, okay? So in here, we are focusing on basically DNA and RNA. And when we have to focus on the elements, basic elements, it is carbon, hydrogen, oxygen, nitrogen, and phosphorus. There is no difference. We have to remember the same procedures for this also. The basic units, they are called as nucleotides. And here they have given you the functions and the importance of it one by one. Okay, let's see. Read it first. Nucleic acids. Can you read from here?
Nucleic acid is the most important molecule out of the main biomolecules in living matter is in genetical aspect.
Mhm.
It is a linear polymer made up of large number of nucleotides. It contains carbon, hydrogen, oxygen, nitrogen, and phosphorus.
Very good, nice. Okay, not only that, though, here they have given you the basic units or the structure of the nucleotide. Better to have this also because this is an extra knowledge one, but you have to remember this, okay? Most of the school papers as well as provincial papers have they discuss or they ask questions based on this also. So better to keep that in your mind. Before that, let's see, go through with the basic properties. Here they have mentioned nucleic acid is the most important molecule out of the main biomolecules in living matter in genetical aspects. Okay, that is a common thing that we can see. When you come to the other ones, nucleic acid is very common. It's the most important nuclei out of the main biomolecules. Okay, when we come to the genetical aspects, what do we mean by genetical aspects? So normally, from generation to generation, the genetical procedures or genetical features, properties come from the previous generation to this generation. From our generation, we are transporting it for the next generation. So that is the genetical aspect. So basically, if someone asks you what is the molecule or the substance that have the ability to transport these genetic properties, we have to call it as nucleic acid. Basically, there are two types of nucleic acids. They are DNAs and RNAs. Basically, this task was done by DNA, but in some viruses, the RNA also have the ability to transmit the genetic features of the properties. And it is a linear polymer. It is a linear one, okay? Linear polymer made of large number of nucleotides. So when we go through the basic unit of nucleic acid, it is nucleotides. Nucleotides. It contains carbon, hydrogen, oxygen, and nitrogen, phosphorus. There are 5 elements: carbon, hydrogen, oxygen, nitrogen, and phosphorus. Those are the main 5 elements that we can see. Let's write down. Nucleic acid is the most important. Write down. Nucleic acid is the most important molecule out of the main biomolecules. 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 In living matter. 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 In genetical aspect. In genetical aspect. In genetical aspect. In genetical aspect. Next one. Next one. It is a linear polymer. It is a linear polymer. It is a linear polymer made up of large number of nucleotides. It is a linear polymer. It's a linear polymer made up of large number of nucleotides. Made up of large number of nucleotides. 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 Nucleotides. Then write down next point. It contains carbon. It contains carbon. It contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. It contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. It contains carbon, hydrogen, oxygen, nitrogen, phosphorus. 5 elements. Carbon, hydrogen, oxygen, nitrogen, phosphorus. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. That are the main elements. Okay, then we'll let's move forward with the next one. Write down. Nucleotide is made of 3 components. We already know that the nucleotide is the basic unit of nucleic acid. So let's see, go through, let's see learn about this nucleotide. Write down. Nucleotide is made of 3 components. Nucleotide is made of 3 components. They are 3 components. They are 3 components. They are nitrogenous base. Write down. Number them. First one, nitrogenous base. Second one, pentose sugar group. Second one, pentose sugar group. Pentose sugar group. Third one, phosphate group. First one, nitrogenous base. Second one, pentose sugar group. Third one, phosphate group. Phosphate group. Those are the main 3 components. Phosphate group. Okay, so take 1 minute and quickly draw this structure. Right, a nucleotide. A nucleotide. Write the topic nucleotide and quickly draw this structure. It is the phosphate group. This is the pentose sugar group. This is the nitrogenous base. Phosphate, pentose sugar group, and nitrogenous base. Quickly draw it. Quickly draw it.
Okay, sir.
Yeah, good. Then write down the row. There are 2 types of nucleic acids. Write down. There are 2 types of nucleic acids. Number them. First one, DNA. There are two types of nucleic acids. Write the first one, DNA. DNA. Write down DNA. It means deoxyribonucleic acid. Write the long term also. DNA, deoxyribonucleic acid. DNA. DNA. Second one, RNA. Ribonucleic acid. RNA. Ribonucleic acid. Ribonucleic acid. DNA, deoxyribonucleic acid. RNA, ribonucleic acid. Ribonucleic acid. Okay. Write the subtopic daru DNA. 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 Write down the function of it. DNA. Write the subtopic DNA. Then write down DNA transfer genetic characteristics from DNA transfers genetic characteristic from DNA transfers genetic characteristic from generation to generation. DNA transfers genetic characteristic from generation to generation. Paramparaven paramparaven. Generation to generation. Generation to generation. Okay. Yeah. Then write the subtopic RNA. RNA. Write down RNA. Write down. The structural unit of RNA is the structural unit of RNA is ribonucleotide. The structural unit of RNA is ribonucleotide. 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 Next point, go write down. Protein synthesis is the function of RNA. Protein synthesis is the function of RNA. Protein synthesis is the function of RNA. Then write down the next point. Significance of nucleic acids. It means important. Significance of nucleic acids. Significance of nucleic acids. Significance of nucleic acids. Significance of nucleic acids. Yeah, let's read it first. Do a quickly read it.
And this one is important in storage of genetic information of organisms. Second one is important in transferring genetic information from generation to generation. Third one is important in protein synthesis process. Fourth one is important in controlling all cellular activities in a cell. The information to control cellular activities is present in DNA.
Okay.
Fifth one is RNA is important in storing genetic information of some virus. Sixth one is the variations occur in DNA due to mutations are important in evolution.
Evolution. Okay, very good. So let's go one by one. First one, important in storage of genetic information of organism. I already explained you when we come to the DNA. Rather than RNA, DNA is playing the most of it. RNA basically playing the important role in viruses. So normally storage is informed. It means important in storage of genetic information. Okay, it includes almost all the genetic information in DNA. Second one, important in transferring genetic information from generation to generation. I already explained you. DNA is the one have that ability. Third one, important in protein synthesis. I explained you in RNA. RNA is important in protein synthesis. Fourth one, important in control all cellular activities in a cell. The information to the control cellular activities is present in DNA. Basically, when we come to our body, almost all the body parts and the functions are controlled by brain. Just like that, when we consider a cell, all the functions and properties, all the features controlled by the nucleus. Then in the nucleus, most important component is DNA. So DNA has the ability to control all the functions of cells. Then the next one, RNA is important in storing genetic information of some viruses. Basically, I told you, when we come to the genetic information or processing or transporting or storing genetic information, DNA has the major role. DNA is playing the major role, but not only DNA, but also RNA has that function. RNA has that ability. That's why they have mentioned RNA is important in storing genetic information of some viruses, basically viruses. But DNA, it is for all the organisms, not only for virus, but basically RNA. But in this point, okay, RNA is important in genetic information storage with viruses. Other than that, the final point, it is very important one. Variations occurs in DNA due to the mutation or informed evolution. Evolution, basically, what did they explain here? With the time, definitely the generation should be changed. If we are just same as our parents, the whole world will be same, because it means almost all the organisms, all the human will be same in throughout the world if there is no any mutation. Definitely mutation should be happen. Because of these mutations, we are not as same as our parents. Okay, there are some sort of different properties, different features, different functions, different characteristic of us. It is up to our own. It's because of the basic reason for that is genetical mutation. So when we come to our next generation, they are not just same as us. They have different properties, different features, different thinking pattern, thinking level, brain capacities. Almost all these differentiation are based on mutation. So from mutation, DNA have the ability to produce different generations. So those generations should have the ability to face the environmental strategies, environmental threats. So if they are able to overcome those environmental threats, සාමාන්‍ය පෙළ විභාගයට මාස පෙනී සිටීමට උත්සාහ කරනවා නම් පෙනී සිටින් බලාපොරොත්තු වෙනවා නම් ඔයගොල්ලෝ තුර ලොකු ප්‍රශ්න ඇති නේද? පාඩම් කරන්නේ කොහොමද? කම්මැලි, අමතක වෙනවා, මයින්ඩ් එක කොහොමද ෆෝකස් එකේ තියාගන්නේ? හරි ස්ට්‍රෙස් එක මැනේජ් කරන්න කොහොමද? මම මේ පාඩම් කරන දේ කොහොමද දීර්ඝව කාලයක් මතක තියාගන්නේ? විභාග ප්‍රශ්න පත්‍ර වලට උත්තර දෙන්නේ කොහොමද? වගේ ප්‍රශ්න ගොඩාක් ඔයගොල්ලෝ තුල ඇති. ඉතින් මම හිතනවා මම මගේ YouTube නාලිකාව හරහා වීඩියෝ මාලාවක් ඔයගොල්ලන්ට ගෙනත් දෙනවා. මේ ප්‍රශ්න වලට ඔයගොල්ලෝ කොහොමද මුහුණ දෙන්න ඕනේ? දරුවන් හැටියට මේ ප්‍රශ්න වලින් ඕවර්කම් කරන්න, ඒගොල්ලෝ මේ ප්‍රශ්න වලට පිළිතුරු සපයා ගන්න කොහොමද කියලා වීඩියෝ මාලාවක් අපේ YouTube නාලිකාවෙන් අපි ගෙනත් දීලා තියෙනවා. ඉතින් රැඳෙන්න එකතු වෙන්න අපේ YouTube නාලිකාවත් සමග ඔබගේ යාළුවන්ටත් මේක ෂෙයා කරන්න. ඒගොල්ලන්ට ගොඩාක් වැදගත් වෙයි. වැදගත් විදිහට පිළිවෙලින් අපගේ වීඩියෝ අපි නිපදවලා තියෙනවා. ඒවා නිවැරදිව ක්‍රමවත්ව ගොඩනගලා තියෙනවා. ඉතින් ඒවා ඔයගොල්ලෝ බලන්න, බලලා ඔයගේ යාළුවන්ට ෂෙයා කරලා මේ අත්දැකීම, මේ එක්ස්පීරියන්ස් එක අනිත් අයට ලබා දෙන්න. ස්තුතියි අපි එක එකතු වුණාට. ඉදිරියට අපිත් සමගම රැඳී සිටී කියලා ආරාධනා කරනවා. ස්තුතියි.